Learning outcomes. After studying this module, you shall be able to know the morphology of hair, how do we classify the hair, the biochemical composition of hair, what are the differences between human and animal hair, functions of hair. Morphology of hair. A hair can be defined as a slender, thread-like outgrowth from a follicle in the skin of mammals. Composed mainly of keratin, it has three morphological regions: the cuticle, medulla and cortex. Each hair shaft also contains a small gland called the sebaceous gland located next to the hair shaft. Sebaceous glands make a yellow, fatty substance called sebum that lubricates the hair. Each time the erector pili muscle contracts, the gland is squeezed and a small amount of lubricant is applied to the surface of the hair. Hair, along with skin pigmentation, is the major natural protection that we have against the sun's harmful ultraviolet rays. Scalp hair also plays an important role in preventing mechanical trauma to the skull. Hair acts as a dry lubricant in areas that rub, such as under the arms and in the groin, and serves to disperse pheromones, body secretions that are involved in sexual attraction. Hair is integral to our body image and can have a profound influence on our self-esteem and self-confidence. There is no other part of human anatomy that can be changed or manipulated so easily. Hair can be groomed, styled, waved, straightened, dyed, braided or cut, and unlike tattoos or body piercing, changes made to our hair can be completely reversed. Hair serves as an important means of self-expression, and the loss of this form of self-expression in those going bald may account, at least in part, for the despair that they may experience. Morphological examination of hair samples is the first step in forensic hair comparisons. The main medico-legal concerns with hair examination include identification of the species of origin, ascertainment of the hair's provenance from the body, and finally, comparison of the control hair sample from a victim to the hair sample from the crime scene. Though it is not possible to definitely identify a sample of hair originating from a particular person's head, unequivocal determination of human origin can be established based on microscopic examination of the hair's cuticle, cortex, medulla, and pigment granules. Human and animal hairs show similarities in having an outer cuticle, cortex, and medulla. The outer surface of the hair is covered by scales. Though there are similar morphological features, the scale pattern provides distinguishing characteristics between animal and human hairs. The scales of an animal's hair show many distinctions such as coronal or crown-like and spinous patterns, whereas in the case of humans, the scale patterns are of the imbricate type, that is flattened. Besides, the medullary index, which is the ratio of the medulla's width to the diameter of the hair, is 1/3 and below in humans compared to greater than 1/3 in animal hairs due to the greater width of the medulla in animals. Hair is commonly encountered in almost all criminal cases involving sexual offences, etc. It may reveal information as species, race, sex, and site of origin, which is very useful from personal identification point. In addition, infectious diseases and morphological and biochemical characteristics of hair help to identify an individual conclusively. Hair being skeletonized tissue are least affected by various environmental conditions as compared to the other bodily tissues. It remains unchanged for a very long period, thus is of great forensic significance. Hair has a distinct specialized anatomical structure. In forensic examination of hair, the morphological features, which are primarily based on the detailed microscopic observations of the three components, that is cuticle, cortex and medulla, play a very important role. But it is observed by various workers that the features from these morphological parameters are not consistent and variations are also very irregular. Now we will study the remaining module with the help of graphics and visuals.
Let us now understand the structure and classification of hair. Hair is a protein filament that grows from follicles found in the dermis or skin. Hair is one of the defining characteristics of mammals. The human body, apart from areas of glandular skin, is covered in follicles which produce thick terminal and fine vellus hair. Most common interest in hair is focused on hair growth, hair types, and hair care. But hair is also an important biomaterial primarily composed of protein, notably keratin. Hairs are generally outgrowths from an organ which is known as hair follicles on the stain. The hair follicle is a tube-like pocket in the epidermis that encloses a small section of the dermis as its base. Hair is embedded in the skin of the mammalian. A typical mammalian hair is comprised of two parts: the shaft, which is producing above the skin, and the root, which is embedded inside a follicle beneath the surface of skin. Let us now understand the section of hair. First is the cuticle layer. The cuticle layer is the outermost layer of the hair and envelops the inner layers. It is tough, dense structure, protects the hair. 6 to 10 layers of flat cells surrounds the cortex. They are arranged in a similar fashion to roof tiles. The outermost exposed edges are open to the tip of the hair. The cuticle layer is colorless and therefore permits the light to shine through. It makes up a good 10% of hair's diameter. Cuticle is important part of the hair which determines the hair species. The cuticle layer is important for the shine and elasticity of the hair. In young hair near the scalp, the edges of the cells lies flat on one another. The hair is only slightly porous and feels soft and supple. The surface reflects the light, giving the hair its shine. The older a piece of hair, the more physical and chemical stress it has been through. The edges of the cells open along the length and at the tips of the hair, or they are worn away. The hair becomes porous and feels straw-like and rough. It loses its gloss because the uneven surface no longer reflects the light evenly. Next is medulla layer. Medulla is the center core of the hair and it comprises of soft keratin protein which may be either present or absent. If it is present, it may be fragmental, interrupted or continuous. It is absent in some species and do not determine the species of hair. Its diameter varies according to the thickness of the hair. In thick hair, it is like a canal and gives the hair its stiffness. In fine hair, it is the only partially there or completely missing. Next is cortex. Cortex constitutes the middle layer, which is made from long thin cells, firmly linked to one another. It provides strength, elasticity, and a unique texture to the hair. The cortex is the thickest of the three layers and makes up about 80 to 90% of hair's diameter. Hardened fibrous cells are bundled together. Every fibrous cell is made up of thousands of protein molecules. These long fibers are twisted together in bundles. The fibrous bundles are surrounded and held together by a mass of softer keratins. The cortex also contains the color pigment melanin, which gives the hair its natural color. White hair has no pigment and contains no melanin. Next, we will learn about the hair bulb. All biological processes and cell division takes place in hair bulb. The hair bulb is bedded in a tight tube of skin, the hair follicle. The slight angle of hair follicle's position defines the direction of the growth of the hair. Additionally, the hair follicle has a straight form for straight hair and a bent form for curly hair. The hair follicle for hair on the scalp or on the beads is 3 mm on average and penetrates into the subcutaneous fatty tissue. The hair bulb is rounded like an onion. The papilla of connective tissue is put over this from below. In the hair bulb around the papilla are two types of cell. The matrix cells known to be keratinocytes as in the epidermis and the pigment cell melanocyte. This is the living part of the hair. Microfine blood vessels, that is capillaries, grow in these near the cells. The life cycle of the hair, the cell metabolism, and particularly growth all depend upon the blood supply from the capillaries. This is comparable to the cell migration and cell transformation in the epidermis. Nutrients are transported via the blood capillaries. Nutrients are transported via the blood capillaries and waste products from the cell metabolism are transported away. Now we will learn about the human hair growth. Hair grows everywhere on the external body except for the mucous membrane and glandular skin, such as that found on the palms of the hands, soles of the feet, and the lips. Hair follows a specific growth cycle with three distinct and concurrent phases, that is, anagen, catagen, and telogen phases. Each has specific characteristics that determine the length of the hair. All three occur simultaneously. One strand of hair may be in anagen phase while another is in telogen phase. The body has different types of hair, including vellus hair and androgenic hair, each with its own type of cellular construction. The different construction gives the hair unique characteristics, serving specific purposes, mainly warmth and protection. Now we will learn classification of hair. First is animal hair. The compositional pattern of the pigmentation can vary widely in different animals. Pigmentation is extreme at the medulla portion as compared to the rest of the areas. Pigmentation is generally found in solid masses called ovoid bodies, especially in animals like dogs and cattle. Hair of animal change its color unexpectedly in banded pattern. In animals, the medulla is far broader than it is in humans. The ratio of diameter of medulla to the diameter of hair itself is known as medullar index. In animal, medullar index is greater than 0.5. Next is human hair. Human hairs have numerous differences from the animal hair, which includes the type of pigmentation, the medullary index, and the type of cuticle. Pigmentation tends to be denser towards the cuticle in human hair. Human hairs are usually homologous in color along the length. The ratio of the diameter of the medulla to the diameter of hair itself is known as medullary index, and in human it is 0.33 or less. Next is differences between human and animal hair. Animal hairs are classified into three basic types. First is guard hairs that forms the outer coat of an animal and provide protection. Next is fur or wool hairs that forms the inner coat of an animal and provide insulation. Next is tactile hairs or whiskers that are found on the head of animals provide sensory functions. Human hair has imbricate scales in cuticle that are flattened and narrowed. Other types of hairs found on animal includes tail hair and man hair, like horse. Human hair is not differentiated and might be described as a modified combination of characteristics of guard hairs and fur hairs. Human hairs are generally consistent in color and pigmentation throughout the length of the hair shaft, whereas animal hairs may exhibit radical color changes in a short distance, known as banding. The distribution and density of pigment in animal hairs can also be identifiable features. The pigmentation of human hair is evenly distributed or slightly more dense towards the cuticle, whereas the pigmentation of animal hairs is more centrally distributed, although more dense towards the medulla. The medulla, when present in human hairs, is amorphous in appearance, and the width is generally less than one third the overall diameter of the hair shaft. The medulla in animal hairs is normally continuous and structured, and generally occupies an area of greater than one third the overall diameter of the hair shaft. The root of human hair is commonly club shaped, whereas the roots of animal hairs are highly variable. Next is human hair classification. Hair evidence examined under a microscope provides investigators with valuable information. Hairs found on a knife or club may support a murder and assault weapon claim. A questioned hair specimen can be compared microscopically with hairs from a known individual when the characteristics are compared side by side. Human hairs can be classified by racial origin, such as Caucasian European origin, Negroid African origin, and Mongoloid Asian origin. In some instances, The racial characteristics exhibited are not clearly defined, indicating the hairs may be mixed racial origin. The region of the body where a hair originated can be determined with considerable accuracy by its gross appearance and microscopic characteristics. The length and color can be determined. It can also be determined whether the hair was forcibly removed, damaged by burning or crushing, or artificially treated by dyeing or bleaching. The characteristic and their variation allows an experienced examiner to distinguish between hairs from different individuals. Hair examination and comparisons with the aid of a comparison microscope can be valuable in an investigation of a crime. Next is DNA examination. Hair that have been matched or associated through a microscopic examination should also we can examine by mitochondrial DNA sequencing. Although it is uncommon to find hairs from two different individuals exhibiting the same microscopic characteristics, it can occur. For this reason, the hairs or portions of the hairs should be forwarded from mitochondrial DNA sequencing. The combined procedures add credibility to each. Although nuclear DNA analysis of hairs may provide an identity match, the microscopic examination should not be disregarded. The time and the cost associated with DNA analysis warrant a preliminary microscopic examination. Often it is not possible to extract DNA fully, or there is not enough tissue present to conduct an examination. Hairs with large roots and tissue are promising sources of nuclear DNA. However, DNA examination destroy hairs, eliminating the possibility of further microscopic examination. Next is the biochemistry of human hair and animal hair. Biochemical composition of hair. Hair is mainly composed of protein, keratin, which is also the primary component of finger and toe nails. 18 amino acids blend together to form a protein called keratin. And among these amino acids, cytokinin deserves special mention as it is rich in sulfur and plays an important role in the consistency of the hair. It is formed by keratinocytes and they are located in the bottom of the dermal papilla, multiply and differentiate. The basic composition of hair contains 28% of protein, 2% of lipids, and the remaining 70% are mineral salts and amino acids. The composition of amino acids of hair is it contains cysteine, glutamic acid, agrenin, serine, threonine, leucosine, aspartic acid, valine, isoleucosine, proline, phenylalanine, glycine, and alanine. as well as some other materials also. Next is the functions of hair. Animal hairs are thick, large, dense, and more prominent in comparison to the human. The more leaves provide protection to animal with sudden changes of temperature. In humans, hairs are less prominent and did not play much role in temperature protection. The location of hair generally indicates its role. Head hair protects the scalp against the burning sun and holds in body heat. Eyelashes and eyebrow hair help keeping foreign matter out of the eyes, and hair in the nostrils and ear canal help in catch dust, debris, and even insects from entering the body. Nostril hairs also helps regulates the temperature of inhaled air before it enters the body. Human body hairs are connected to touch receptors in the skin that allow us to feel and, in that sense, collectively serve as a protective warning device. Hair also serves aesthetic function of adding beauty to the human body. Head hair frames the face just as eyebrows frames the eyes. Hairstyles often serves as a form of individual expression. Facial hairs helps differentiate man from woman, and pubic hair indicates that a man or a woman has reached puberty and is becoming sexually mature and ready to reproduce.
Now let us summarize what we have learnt in this module. Hairs are generally originated from an organ which is known as hair follicle. A typical mammalian hair is divided into two parts: the shaft, which is protruding above from the skin, and the root, embedded inside a follicle. Animal pigments are often found in solid masses called ovoid bodies, especially in dogs and cattle. The ratio of the diameter of the medulla to the diameter of the hair itself is known as the medullar index. If the medullar index is 0.5 or greater, the hair is of an animal, and if less than 0.33, then it is derived from human. Rodents and bats have a coronal cuticle with scales that give the appearance of a stack of crowns. Cats, deer, seals and mink.